Good morning. My name is Alexey Aksimentiev. I am one of the PIs of the NIH Center for Micromolecular Modeling and Bioinformatics, the one that is co-organizing this workshop. The plan for today is for me to give you 2 lectures on 2 related subjects, and then you will have a choice of 2 tutorials to work in the afternoon with my TAs, Chris and Jim. Could you stand up, please? Yeah. So here they are. Thank you. So basically, my research area is probably DNA in all its manifestations. So my first lecture will be about DNA systems, and the other lectures will be about modeling the interface between bio and nano, where again it will be focused on DNA systems. So in a way, DNA is probably the key word for today. How many of you are already familiar with molecular dynamics simulations? Quite a few, and I mean beyond the lecture that you had 2 days ago. So some of you, okay. Because that's kind of important for me to understand in how far in the details should I go in my lecture. And also, I would really appreciate if you just ask questions as I go, because otherwise I will be just talking and talking and talking, and I will get tired and bored. It's much interesting for me if I get some response from you. So let me start, and again, please feel free to interrupt me at any time. So I am coming from a physics department, and in physics we like formulas. And as many physicists, after a long and extensive career, that's not me, but other people would ask a question, what is life, right? So like this guy, Schrödinger, who wrote a famous equation. He also wrote a fairly famous book, What is Life? So he was interested in determining what life is. But from a physics point of view, the ideal answer would be a formula. You know, here's an equation of life. And of course, that's not possible. Although I like to think about DNA as the answer to that question from a physics perspective. So in a way, DNA is a formula that describes life, or can be used to describe life, or that's what life is used to describe itself. You can put it also that way. So many of you are familiar with DNA, probably all of you. It's difficult not to find its double helical logo somewhere. But basically, DNA for me is a blueprint of an organism, and it is organized in a cell at different levels. So most frequently we talk about DNA and we think about its double helix, which is shown here. And we are all familiar that the DNA has a sequence, right? The nucleotide sequence of DNA, G, T, A, and so on. So that's the sequence. On the other hand, we are also somewhat familiar with chromosomes, right? So chromosome is something that can be visible in a microscope at a special time in a cell cycle. And chromosomes, that's also genes and so on. So basically, that's kind of the higher scale, and that's the lowest scale. But in between, there is all kind of structures in which DNA organize. Going through chromosomes, going down, you basically have a fiber that is a thick fiber, and then a thinner fiber, eventually going to the beads on a string and so on, and to the molecule of DNA. So DNA organizes in a cell in multiple levels. And very frequently, in biology, when people ask about or talk about DNA, they mainly are concerned with the DNA sequence, not so much with its physical properties. So let me tell you give you a precise definition of what DNA is. So DNA is a polymer, and it is made of nucleotides. So a nucleotide is a building block of a polymer. So many of those nucleotides would make a long polymer. Nucleotides are shown here. So typically, you are familiar. I mean, you most likely are familiar with double-strand DNA. So it's shown here in this representation, where each of the strands is shown in different color. Now, when you put it under stress, like high temperature or special solvents, DNA would split into two single strands. So that would be a single-strand DNA. So single-strand DNA is a polymer, and double-strand DNA is made of two such polymers that are joined non-covalently. So each polymer, single-strand DNA polymer, is made of nucleotides. So here they are. It's like ladder-like, and each nucleotide itself has three functional groups. We can talk about the backbone group, which includes a sugar ring and a phosphate and the base. So the base is where the sequence of DNA is written in. So here's a little bit more details about the DNA. You're looking at DNA structure in the double helix. Of course, you know that there's a complementary base pairing between A and T and C and G nucleotides. So they come together and form a helix. Now, what I like to stress here is that the difference between different nucleotides is very small. So we're talking about a few atoms. So in that sense, the molecular blueprint of life, so to say, or the DNA code is written in atoms. So keep that in mind. So the DNA, a human DNA weight is about 10 picograms. So basically, if you take human DNA, like one copy of human DNA from every human on Earth that is currently living or has ever lived over the last 10,000 years or 100,000 years, that would still be less than a gram. So you can think that kind of contains information about our civilization in a way. All right. I've been talking for 10 minutes without any questions. I'm getting... let's do this. So this is a central dogma of biology, right? First came the DNA and then the protein. So the DNA contains information how to make something other than DNA out of it. Typically RNA. So RNA is made from DNA by an RNA polymerase. Okay. And that RNA is then transcribed into a protein by a ribosome. So that's the central dogma. But that is not a complete story. DNA can be also transcribed into RNA, and that's it. End of story. Actually, most of the human genome does not code for proteins. Only 1% or 2% code for proteins. The rest is just there. Of that dark matter of human genome, 50% codes for RNA that is used in all kind of regulatory processes. But from a biologist's point of view, that really is the most important thing, the role of DNA. So it just carries a sequence. But that is really a kind of simplified view. In fact, there are compelling evidences that the physical properties of DNA are as important as its sequence. Well, first of all, the DNA has to have a certain properties that permit readout of its sequence by DNA, RNA polymerase, it has to come together in the form of two complementary strands, and it has to come apart when the conditions require that. The DNA has to be able to wrap around histone proteins and yet be stable over over the entire period of organism development. So in a sense, DNA is not a random molecule, I would say. So when you look at the physical properties of DNA, they're fairly unique. For example, if you actually, I'll show you this in greater details. But basically what I'd like to stress here at this slide, that the physical properties of DNA underlie its functionality. So over the past 12 years, I've been working in this area using molecular dynamics as a kind of a microscope. So Klaus Schulten has given you probably a fantastic lecture on that subject, so I'm not going to repeat it. What I'm going to do, I'm going to show you how one can use molecular dynamics simulations to study DNA systems. Okay? So you will have one tutorial that is specifically designed to introduce simulations of DNA systems, actually DNA protein system. And what we do with our tutorials in my group is that we basically take a snapshot of a research project. So for the case of DNA protein tutorial, that was a research project that Chris did. And at some point we said, okay, it's almost done. Let's write a tutorial. And in this way we preserve our know-how, so to say. So when a new student comes, he or she can just start where the project left. So in that sense, the two tutorials that we will be giving you today are kind of, I would say, expert maybe. There are some parts that are fairly easy, but basically because it is a snapshot of our research project, no matter how we try to simplify it, it is something that you can basically publish. Okay. So in that sense, it's a little bit different than say, MD tutorials that you had, which was basically to get you a feeling what molecular dynamics simulation is. Here, it's more like what is a research project using molecular dynamics simulation is. So in both tutorials, you'll learn how to build DNA, and after you build DNA, you'll learn how to set up variants of those systems. So here's some projects that are going on in my group. We are looking at DNA assembly in chromosome at different levels. We are looking at the interface of nano and bio. This is a pilot project sequencing DNA using a nanopore. In this case, this is graphene. And we also are interested how DNA is repaired and replicated. Is there any questions so far? All right, let me give you a few examples. So here's the first one. Stretching DNA. So DNA is an extensible molecule. I think that was the title of one of the famous papers on this subject. So it is possible to take a single molecule measurement approach to determine the mechanical properties of just one DNA molecule. How many of you are familiar with single molecule measurements? Not at all. All right. Okay, so here is a crash course on single molecule experiments. In this particular case, you are looking at a setup called optical tweezers. So what you have in there are two beads. Those are typically micron-sized beads. they are coated with special molecules, biotin or streptavidin, I don't remember which one of them, and typically they are floating in solution. All right. Then you have two instruments that you can manipulate with the beads. In this case, one is a micropipette, so that's like a pipette, but that has a very small tip. And what you can do with that instrument, you can apply suction and trap one of the beads. Of course, there are many beads, but I'm showing you only one. What you do prior to that, you add a DNA molecule to the bead. And how do you do it? So you chemically modify the ends of the DNA to contain a molecule that would interact with the other molecule that you coat the bead with. So like streptavidin, biotin. So they come together, and if everything is done right, you'll have only, well, you probably will have more than one, but only one of them would make contact with the other bead. So the other bead is placed in the focus of a laser light. So that's schematically shown here. So it is a physical phenomenon that when you focus light, a special kind of light on a special a light that has a special intensity profile on a micron bead, it will be trapped, meaning it will not be able to escape left, right, or up and down and in the page. So you create an optical trap, and the idea here is that you can attach one end of the DNA molecule to the optical track and the other end of the DNA molecule to the bead. And you have a microscope, and you can see it because the beads are quite big. Well, they are microns, but you can still see it in an optical microscope. And what you do next, you just either move the stage, meaning the pipette, or you can move the focus of the light, and this way you can change the distance between the beads. So as you change the distance between the beads, you will apply a force to the DNA molecule. So you'll never see the DNA molecule in this experiment, although you probably could if you had some kind of intercalating dye, but most of the time you don't. You see the beads, but you know that the molecule is there. And how do you know that the molecule is there, and how do you measure the force? And that is done the following way, the clever physics way. You look at the bead, and you look how much it fluctuates. So remember, so I recently been to a party, my daughter's friend party, and they have giveaways those helium balloons, you know. So helium balloons are on a string. So think about it as a helium balloon. So helium balloon on a string, and it fluctuates, right? The amount of fluctuation by the helium balloon, or in this case the bead, will determine how the tension in the DNA. So basically, if you look at the fluctuation of the bead, from equipartition theorem, you can compute the force with which it's scattered. So that's how you get the force. Okay. So in this setup, you can get the extension and the force. And what you can do, you can measure the force extension curve of DNA. So that's what is shown here. Force and fractional extension. In this case, it's fractional because it's compared to the length of the contour length of double-strand DNA. So you can see that single-strand DNA and double-strand DNA, they have fairly different physical properties. So if you look at the single-strand DNA, the force increases gradually as you extend it, and then eventually it rapidly increases. Whereas for double-strand DNA, the behavior is different. For double-strand DNA, the force increases very slowly, but then it increases very rapidly, and then there is this overstretching transition, plasticity transition. So have you ever tried to rip a plastic bag? Who has done this experiment? You know, you pull it, but then eventually it yields, and then woo. So that's a plasticity plateau, and that's what happens with double-strand DNA. Okay, and And I guess that's why it was a science paper, because it was boring like that. Maybe it wasn't a science paper. But when something happens interesting and unexpected, you get a high-profile publication, I guess. All right. So overstretching plateau, what is it? Yes.
What is the wavelength of light you used there?
What was the wavelength of light?
Yeah.
Typically, the one, yeah, it's a visible light. I don't know exactly which one it was.
Okay. And how big is the single DNA molecule?
It's in this case, it's 47 kilobases, or it's tens of microns, basically. Right. So you can see the separation between the beads quite precisely. All right. So the overstretching transition is something that could potentially be investigated using molecular dynamics simulation. Okay. And that's what we did. So let me give you two examples how to do it. The simple one, the easy one, and the difficult one. Okay. And of course, the difficult one is the one that you should do. Yeah. The easy one is this one. It's called SMD, Steered Molecular Dynamics. Did Klaus talk about steered molecular dynamics? All right. Easy one, because you are guaranteed to, it's one of those, I love steered molecular dynamics because it is always successful, okay? You are guaranteed to see something because the way it works. So you fix one end here, and you have a template atom, or like a template spring that is attached to the atom, and you move it with constant velocity. So you predetermine what will happen in your simulation, meaning you will stretch it no matter what, because if you choose the proper velocity and stiff enough spring, you will see something. So in this case, we are stretching a molecule of single-strand DNA, and yeah, so it goes like that. So nothing really spectacular here. It stretches. And of course, we know the force and extension, so we can plot the force extension curve. So for single-strand DNA, it was also measured using AFM probe. And here's a typical force extension curve of that you get from AFM. Although this paper is still kind of controversial, there's this plasticity plateau in single-strand DNA that some people see, some people don't see. But if you do a molecular dynamics simulations, it's steered molecular dynamics, you just pull on it. This is the kind of data that you'll get. So first of all, do you know why it goes like that? No? That has to do with the spring. So basically, you have, the spring fluctuates. The amount of the jiggering is determined by how stiff the spring is. That's why it looks like that. But basically, if you average it out, you can see that we get some kind of a plasticity plateau over here, right, for single-strand DNA. And if I play the movie back, not back, again, you can kind of tell where it's coming from. See, the bases are stacked, and now they start breaking apart into groups of 3 or 4. Yeah. So that is a molecular cause of the plasticity plateau. So you're not stretching the DNA. You're not stretching the backbone. You're breaking the base pairs. So here's the outcome. So you could say, yeah, great. We basically reproduced experiment. And to a certain degree, we did. We stretched DNA, and we also observed the plasticity plateau. Of course, if you want to compare the numbers, it becomes more difficult. And that's basically the biggest drawback of steered molecular dynamics is that typically, well, you get what you paid for. You end up with forces that are considerably higher than the forces that you typically measure in an experiment. And the reason for that is, of course, the time scale. Right, that was probably 30 nanosecond simulation, and in experiments it takes seconds. So you're accelerating processes, and not surprisingly, you get higher forces. Do you know why you get higher? Yeah.
So for that plot, I'm trying to guess how to
Right.
And the other is just the length comparing with the initial point, so indicating
Yes.
the change.
Right.
Interesting things. I mean, you asked the question yourself, but still it seems puzzle me. It seems like at one point, at one axis, it has multiple faults associated with that.
Right. Yeah, well, it could have been that it's actually this LZ is the position of the template. You know, you move the template, and for each position of the so as a function, so everything is done as a function of time. Okay. So this is something that you don't get directly from a simulation. What you get from a simulation is the force versus time. All right. And you also get the position of the end of the DNA versus time. Now, the position of the end and the force, they are correlated, but not precisely because, you know, there is some kind of a back and forth motion. And that's basically what you see here. So it's a, think about it, this is a parametric plot. So you had a force versus time and position versus time. So your time is a parameter, and then you'd make a parametric plot of one versus another. So that's how you should interpret this. All right, so the problem here is that your forces typically are much higher than they should be, which is actually expected because you accelerate processes. So typically, when you have a molecular bond, so here's one of the things that I'd like you to remember from my lecture. When you have a molecular bond and you apply a force, the probability of it rupturing is an exponential function of the force, right? So you increase the force by twofold, the probability of something breaking increases e to the 2, right? You increase the force by tenfold, and the probability is e to the 10. So that's basically why with steered molecular dynamics, although you are guaranteed to see something, because you're breaking molecular bonds, typically you end up having higher forces. So we used a different method to accomplish stretching of double-strand DNA. We also have done it for single-strand DNA. And you can actually do it with the system that is described in the tutorial for single-strand. So we did something different. We said, OK, we are not going to do steered molecular dynamics, partially because other people did it before us, and partially because we thought that this is a superior method. So what we did was the following. We took a molecule of DNA, in this case, double strand, and we linked it to itself through periodic boundary conditions. Periodic boundary conditions are typically used in molecular dynamics simulations, although I think the MD tutorial doesn't really introduce them. So our tutorial does introduce periodic boundary conditions and pressure equilibration. And in this case, basically this end of the DNA is equivalent to that end. And you can create covalent bonds in cross-periodic boundary, no problem. So we have a patch to do it in the tutorial. And then you'll end up with having effectively infinite DNA molecule. Okay? So that was one thing. Second thing is, how do we stretch something that is effectively infinite? And that's where we mess with a with a barostat. I don't know if did Klaus introduce pressure control? Right? So pressure control works by scaling the size of the system in x, y, and z independently, in principle. So what we can do, we can set the target pressure in x, y, and z plane to different values. So in this particular case, we said, OK, well, let's have pressure pressure on the surfaces of the system that are normal to the DNA to be 1 bar. But let's have a negative pressure in this plane. So negative pressure means that the system would want to expand. And that's exactly what happened. So the system would want to expand, and it will stretch the DNA by deforming the simulation system. Let me show you a movie that illustrates how it works. So you'll be looking at this one. So it is torsionally constrained, meaning that the end of the DNA is connected to itself. But you can see how it stretches in this case. So first of all, it has time to rotate around and explore all the conformational degrees of freedom before it starts to pop. So you can see that one domain of the DNA breaks first, and the other domain of the same sequence breaks second, and then the other fragments of the DNA would break. So that is something that is, I would say, more difficult to set up compared to SMD. SMD is very straightforward. This one, you need to set the barostat to different coordinates, but it's not really rocket science. It's fairly straightforward. If you are interested in doing this for whatever system you have, just ask me, email me. I know people are interested in applying it for unfolding proteins. That could be a good idea for something that is like a fiber. So in this particular movie, you saw that the way the DNA broke was by breaking this base pairing, by breaking base pairing in steps. Now, if I introduce a nick, so a nick is a break in a covalent bond, which corresponds to torsionally unrestrained DNA, you'll see the following. So again, this is using our anisotropic pressure control, so it's effectively infinite. But in that case, as DNA is stretched, it also unwinds. So you'll see how from having 2 turns, it will end up having just 1 turn, and then, maybe half a turn. Right. So it makes a big difference when you compare the makes a big difference whether it's torsionally constrained or not. And the forces that we get from those simulations are actually in good agreement with experiment. For example, for this one, we've got about 110 piconewtons, and it should be around 100. And for this one, it should be around 60 piconewtons, and we are getting around 75. So we are not like a factor of 10 off. We are still a little bit off, but that could be just the issue with the force field, not so much with the method. Okay. Is there any questions to this part? Yes?
Do you get the force by integrating the pressure over the surfaces, the perpendicular surfaces?
Yeah, so how do we get the force? That's an excellent question, because we don't measure the force directly. Yeah, so what happens with during the simulations, the system, so it used to be like that, it will shrink. So basically, you just compute the area. There is a simple, it's like a simple math. You know the tension. And yeah, so basically, pressure is force per area. So once you know the pressure, you multiply it by area, and you get the force. Yeah. If there are no questions to this, I will move on to a different subject. Yes?
I have a crazy question. So that simulation shows, double strand DNA stretching, right? But I just wonder whether you or any people have even tried to simulate, like, you just threw the 2 single strands in the system and trying to see whether the 2 strands come beautifully folded into, a double strand DNA.
Yeah, that is right. That is the most common and the most interesting, probably, process. And people have tried and still trying. It's very difficult.
That's exciting.
This is a very, very tough process to simulate with all atom, specifically because it takes a lot of time. So even for the simplest system, the hybridization timescales are in tens of microseconds. So maybe using Anton, one can do it. People use different methods to simulate hybridization. One of them is replica exchange, where you have multiple systems at different temperatures, and you kind of swap them. That's my personal opinion. I don't think we have a good example where one successfully modeled hybridization process. There are some recent studies, successful ones, where people simulated folding of a short hairpin. So what a hairpin is just a single strand that can fold and form a duplex. So if you have a hairpin made of 6 nucleotides, you can see it folding. That was done, but no more than that. Yes?
Does the molecular dynamics force field actually allow the backbone to ever break? Is there some force that can be applied that would actually overcome the...
No.
Right.
No.
So
But
how does that play into the like elastic solid between the DNA?
yeah, so right. So if you look at experimental force extension curve, it ends somewhere, right? It's not just that we cut off here the graph and here it goes. No, no, no. So in experiment, what happens eventually, the DNA dissociates from the bead. It never breaks actually in experiment. I was looking for it. At which force would the DNA break? Like actually single molecule measurement. It's very tough because typically something else breaks, because all of the it's not covalent bonding or it's something like that. So for all practical reasons, you should not be worried about it. That's not the biggest problem. Of course, in MD it cannot break because of the way the force field is made. All right, let me give you a different, let me tell you a story about a different physical process. You can tell I use physics a lot because we've just finished renewal where physics. So don't be surprised about that. Okay, so same charge, same charges of same kind, they repel, right? And different charge, electrical charge particles attract. Everybody knows that. Yes? Is it always true? Is it true that the same sign charges always repel? Yes? No. Wrong answer. Same sign charges can attract. It depends where they are. So in vacuum, yeah, they repel. But once you put them in a medium, everything can happen. They can repel, then they can attract, and then they can repel again, because the medium would change the interactions. And DNA is not an exception. Well, think about it. DNA is actually highly, highly charged. I think I forgot to mention that. But basically, DNA, one nucleotide carries 1 electron charge. So maybe 1 electron charge is not like a big number, but that's per nucleotide. So once you get many of them in a chain, you'll get a highly, highly charged object. And so how is it possible that this highly charged molecule forms compact structures such as chromosomes? How does it pack inside a viral capsid? How is it possible? Because it should self-repel and never come together to form anything compact. And the reason for that is that DNA is not in vacuum, it's in a medium, and the medium is electrolyte solution. What is electrolyte solution? It's basically salt dissolved in water. And because of that salt, or depending on what kind of salt it is, the DNA will have different properties. It is possible to observe effective attraction between DNA molecules for when it's surrounded by counterions that have charge greater than 2. That's an experimental fact, but let me show you how we can study it using molecular dynamics simulation. So I would consider it like extremely simple brute force molecular dynamics that we can now do because we have access to good computers, or let's say because the computer science evolved and we have computational facilities that doesn't require us to scratch our hand and come up with some kind of fancy methods. This is brute force MD. What we do here is we take 2 DNA molecules. They are effectively infinite in this direction. We place them in a box with water and ions, and then what we do is we apply a harmonic potential to the center of mass of the two molecules. So harmonic potential is there to keep the two DNA molecules at a certain distance, right? Of course, they will not be at that distance because they interact. So what will happen, the molecules, they would also want to go away or closer to one another. But we have the harmonic potential that will enforce the distance. So the DNA will displace a little bit, but then the force in this virtual spring will match the interaction force between DNA. And that will give us the measure of the effective force. So really, brute force approach, what we do, we just run it and get the force. So here's a movie that illustrates the process. So in this case, we fix the distance at 30 angstroms, center of mass. And within the time scale of the simulations, which was not much actually, it doesn't work anymore. Yeah, here we go. So the DNA is able to explore all possible conformations, meaning it could rotate around its axis. It could also displace. And what is not shown are water molecules and ions that move around. So basically, once you get to the time scale of hundreds of nanoseconds, let's say, you basically explore all of those degrees of freedom that are very difficult to take into account in the theory, so all those entropic terms. So you can expect to get the force that is correct. And that's what we did. So we could model it. We could run the simulations at different distances and obtain directly the measure of the effective force between DNA molecules. Versus distance for different electrolyte conditions. So sodium, which is sodium ions have charge of +1. We always observe repulsion, and for magnesium ions, which ion has a charge of +2, we observe some kind of attraction. All right, so that's a direct measurement of the force, but what did we learn from it? And what we learned from it, which perhaps is the most important, is the mechanism. What we learned from molecular dynamics simulation in this case is the mechanism, or in other words, an answer to the question, why do they attract? Why do they attract? And the reason is that a magnesium ion would form a very peculiar conformation between at the point where minor groups of DNA form contact. So it's like a double handshake. So I am a magnesium ion, and I form a contact with one DNA and with the other DNA, and that's how I bring them together. Yeah. So that's how you can use molecular dynamics to measure effective forces. So we have done it in 2008. We also work with experimentalists that use magnetic tweezers to twist DNA and form plectonyms. And basically what we did, we measure again the effective force. And in general, we observe a good agreement between the forces that we predict in our molecular dynamics simulations with the forces that were inferred from single molecule measurements. Although I must say here that actually in this particular measurement, the forces were not measured directly. What was measured just the plectonym formation. So let me move on and show you some other applications of molecular dynamics. Are there any questions about DNA and DNA interactions? Okay. So this slide is here to embarrass Chris. No. All right. So DNA is actually a very interesting subject. So not only DNA molecules can attract side by side, they can also stack. And that was discovered in this paper that was published in Science, basically where they took a bunch of short duplex DNA vigorously shake it, and then they observe formation of liquid crystalline phases, consistent with the notion that the DNA stack end-to-end like that. And once they stack end-to-end, then of course the question was maybe that's how we end up having DNA as a carrier of genetic information. So what we could do, and I like this project because it illustrates the range of MD protocols that one can use to study effective forces. In this case, end-to-end DNA interactions. So here's the first simulation that Chris did, where he just took 2 DNA fragments, again, placed it in a box of water ions, and what is not shown here is that the DNA molecules were constrained to move along one axis. So how do you do it in practice? Well, you just apply some kind of restraints that would constrain the position of the phosphorus atoms of the DNA, so they are on a cylinder, so to say. So the DNA can still, rotate around, can move up and down. Everything else is free to move, but they are effectively set to collide. If they come together, they will form a bond. And that kind of simplifies this kind of probing of the interactions, because if I were just to let it go, they would probably rotate, and it would take some time to actually observe them collision. So for exploratory purposes, this is perfect system, because look what happens. The DNA would just come and get stuck very rapidly. So what we can do now, we can perform the same kind of experiment. This is really a computational experiment because we didn't know the outcome of it beforehand, using a different kind of chemistry at the ends of the DNA. So when you cut the DNA, you have a choice. You could have a phosphate. You can leave or remove the phosphate at the end of the DNA. So it's like a technical details, but typically when you set up a DNA molecule simulation, and if you've done correctly, the charge of the DNA should be the number of nucleotides minus 1, because typically you would remove the phosphate at the end. But there are some biochemical processes at which you end up with a phosphate. So now if you repeat our simulations, you could see that the terminal chemistry determines the state in which the DNA molecule ends up. So you see when the phosphate is absent, the DNA molecule can kind of misalign, whereas in this case, and I can almost hear it click, it forms a perfect double helix. So really now think about it. If like a lightning strikes or an enzyme comes in and facilitates this formation of the covalent bonds, then you'll get a DNA molecule that is twice as large and so on. So you could see how it could potentially be involved in the origins of life, but again, we have no proof for it. But anyway, so that kind of illustrates you how you can explore interactions between DNA through molecular dynamics. Now, of course, you can say, but didn't you restrain the DNA molecule to move along one dimension? And didn't that predetermine the outcome of the simulation? So on the one hand, yes, we did. On the other hand, if the DNA interaction was repulsive, we would see it immediately from our first set of simulations. So it's always good, and I always tell it to my students, if you have in mind a method that is probably not super accurate, but will give you a fast qualitative result, do it first. Because you can spend lots of time using some kind of sophisticated method and then not seeing anything. Because typically, if you see something at the qualitative level, if you do a quantitative simulations, well, you'll just do it better, but it probably won't alter your result. So in that sense, the first simulation that we did was good, but now to convince ourselves that this is real, we had to go through a bunch of high-end simulations. So this one is not a high-end simulation. This is just brute force simulation in which we start with an assembly, and we're just curious if that assembly will ever fall apart. Okay, you can ask a question like that. I don't recommend, unless you have spare cycles somewhere, which we had, and we just had to run it. So And what you see is here that the DNA molecules, the two of them, they stay together. They don't break. Sometimes you can see they can even twist around without breaking the bond. So that told us that probably if we keep doing this, we are not going to see much. So that was almost a microsecond simulation, which was done some time ago. So the next thing that you can do now to learn something about the energetics of the interaction is that you can use stirred molecular dynamics to break the bond. So stirred molecular dynamics, again, is used to pull two of them together. What you see is that it gives you the mechanism, how it dissociates. Because if you compute the force from it again and try to do the free energy calculations through some kind of Dzerzhinsky method or something else, you're probably not going to get something quantitatively accurate. But what that simulation showed you was the mechanism. How do two molecules come apart? How does the rupture happen? So the molecules, they would tilt and slide off. Okay. Finally, one can do a more sophisticated calculation, so-called umbrella sampling. Has it been introduced, umbrella sampling? Okay, so I'm not going to introduce it. I'm going to tell you what it is. And believe me, it's true. That's what my daughter always says. So you're looking at two ends of the DNA. And what you do is, it's kind of similar to what I already introduced. But basically, you have a spring, harmonic spring, and you hold it. You use the harmonic potential to hold the distance between the two DNA ends at a certain distance. And you do an ensemble of such simulations. So typically, tens of such, 20, 50. And you choose your spring such that when the distance fluctuates here, it overlaps, it explores the same region as the neighboring one. So basically, these fluctuations, let's say they typically are set at 20. But the distance sometimes would go to 90 and sometimes to 21. And the next bin would be set at 20.5. And again, the force would be such that the distance would go between 19.5 and 21.5. So you'll get overlapping distributions of this end-to-end distance. And then, if you've got it, some very kind people have developed a method to extract the potential mean force or the free energy between those two ends versus the distance. So there's a method. And believe me, it's true, it's working. But basically, once you get those histograms, you can use the method and you get this, which is the free energy versus distance. This is very general, gold standard method for obtaining free energy versus some kind of reaction coordinate. So we've just applied it here. And so what can we learn from it? First of all, it should be flat away when the two molecules are away from one another. Why? Because the interaction is short range, so when the two are far away, the free energy should be flat so that the gradient of that, which is the force, is 0. Right? But then we can see from the shape of this potential mean force that it's very short range, and we're also getting some kind of a quantitative estimate of what the free energy of interaction is. In this case, it's like -6 kcal/mol. So that's if you if you are interested in free energy calculations, that would probably be the first method that you'll encounter. So we've got this number, but we were not happy with it because it was kind of big. And also when you do this free energy calculations, you have to be careful because you apply restraints. In this particular case, we applied restraints to hold the DNA molecules. And restraints affect entropy. And so in a way, the values that we got is correct, but only for the processes that we measured. So there is a different method called standard binding free energy calculations, which is really high-end. It's probably as high-end as it gets, and we don't have a tutorial on it. Probably will never have. But it's fairly difficult. calculation. I'm just showing it here just to show that it's possible to do it. And Chris did it for those 2 molecules of DNA. So basically, the question is the following. If you start with 2 DNA molecules far away, what is the free energy difference between them being very far away and forming a bound state? To do a calculation like that, you need to split it into a path. So in this case, we split it into 3 or 4. So the first question was, how much energy does it take to bring 2 DNA fragments from infinity to something that we can simulate? Something that we can simulate is a distance of, let's say, I don't know, 5 nanometers apart, right? So that kind of calculation you can actually do assuming that particles are just spheres. So you can do screen Coulomb interaction, and it will be fairly accurate because particles are still far away. So once we brought them to the range of our all-atom simulation, we can ask the following question: how much energy does it cost to align them, right? So they're not just randomly aligned, but to orient them end to end. That will have an energy cost, and we can compute it. Once we've computed that, then we can ask the question, what's the free energy cost of bringing them together? And that we've done. And then the final step is we ask the question, what is the free energy cost to letting them go? So once we compute individual components, and assuming you've done it correctly, we can add it up and get the number. Okay? And that's basically what this calculation is about. If you are ever interested in doing this, please ask us. We know how to do it. As we were doing this calculation, we were worried that maybe it will not work out. It's a fairly difficult calculation, and I'm glad it worked out. But we had a plan B. And I think the plan B kind of calculations you'll see more and more and more. And if you are serious about going into this field, I think you should try it because I think it's awesome. So what is that? We wanted to, well, first of all, we wanted to compute the free energy of binding. And what is the free energy of binding? So typically from a biochemistry book, you would say that if you have a mixture of component A and component B, and you are interested in the free energy of binding between A and B, all you need to know is to find the concentration, right, at which the concentration of bound and free molecules is equal. Something like that, like chemical equilibrium. So that's what we set out to do, but in all-atom simulation. So you're looking at a system that has around 500 fragments of DNA molecules. And this is atomistic simulation. So water, ions, everything is there. It looks fairly dense. And here's the simulation. To our surprise, we could see those fragments forming long aggregates within the time scale of our simulations, which was just 250 nanoseconds. Now, 250 nanoseconds may not seem long, but we have 500 fragments, so we have we have good statistics. So you can multiply 250 nanoseconds times 500, and you are already in almost 100 microsecond range. So from that simulation, we can estimate the k-on rate, meaning how frequently the association happens. We could also estimate how frequently the bond between 2 DNA molecules breaks. So we can get k-on and k-off rates directly by looking. Yes?
How many fragments in the system, and how big each fragment is?
So each fragment was 10 base pairs, and we had, I think, 460. Yeah.
416 fragments.
Fragments, yeah. So it's, I would say it's a big simulation. But there's nothing about the simulation itself that is, so to say, fancy, right? You just have to build it and run it. I think most of the problems with this particular case was analysis, because you have lots of fragments and you want to classify which contacts you consider as contacts and which you don't. So the actual analysis part, I would say, is the most difficult part, assuming that you have access to computers that you can run it, which you probably now do, yes?
How many processors and how many do you need to?
This was actually done in 2010, and we were very fortunate. We had access to XE6 Cray machine, and it wasn't that long. I think it was maybe a week of a calculation, maybe 1,000 CPU, maybe 2,000. So it wasn't something. It wasn't something you cannot do now. Like you can probably. I mean, at that time it was maybe very high-end simulation, but now probably majority of people can do it. So it was just $1.5 million. Yeah, but basically my point is, so I call this approach observational thermodynamics, because you prepare the system, you run it, and then you count how many times the bonds form, how many times the bond break. So you get K on rate, K off rate. You can divide one by another, take a log of that, and you get the binding free energy, which was actually pretty good which was in a good agreement with the estimates that we did using this fancy method. Okay, so I'm not against fancy methods. It's just sometimes, if I can do something simpler, I just do it simpler. So that's basically my approach. Is there any questions to this part? Yes.
Did you see any dependence on the length of the fragment? So say if you have already attached, is it faster to attach a third one?
I don't think our assumption was that no, that it's independent. At least that's what we did in our analysis. I don't think we actually look for it. What? Yeah, we didn't look for that, but I think if you are in a regime where there's still lots of single fragments, probably doesn't matter. But once you go to bigger ones, then yeah, probably. Yeah.
So you did a study where you had two infinitely long chains, and you noticed that the magnesium mediated the
Right.
interaction between the DNAs. When looking at end effects here, where the DNA is a finite length, did you consider or look at the location of the magnesiums along the length of the DNA? Perhaps they were more likely to be found toward the end or toward the middle. How that affected the.
Yeah. Well, that one was done without magnesium.
Okay.
And the reason for that was that because experiment was done without magnesium. Although experiment was later also done with magnesium, and then it becomes very complicated because you have competition of end-to-end and side-by-side. Yeah, it's a simulations that can be done. We haven't done it. I should also say that this is also the point to emphasize or stress that with molecular dynamics simulations, you typically have two problems. One is a time scale, and the other one is a force field. Okay? So I'd say that right now we are in this regime where probably time scale wise, there's still many problems that we can actually assess, that we will have adequate computational resources to get the number right. For example, this one. We should be able to get the number right. Now, the force field is a different issue. So there are limits to the force field. And in this particular study, our energy of interaction, this is actually quite high. It's about by 2 kilocalories per mole higher than what's measured in experiment. And that probably has to do with the way the force field describes the end-to-end stacking. Nothing is perfect, and the force field has been developed 20 years ago when 300 picosecond simulation was a really long one. And since then, basically, development in the software and computer infrastructure outpaced development of the force field by far. So basically what happens now, suddenly everyone can run those long simulations, and issues with force fields start to come out. Because finally we can compare the simulations done with the force field with something that someone can measure, right? And sometimes it works, sometimes it doesn't. So one also have to be careful with interpreting the simulations, because force field can have a bias. In particular for RNA simulation. I think for DNA simulations we have a set of force field combinations that works. And that's basically recent because we've worked a lot with single molecule people trying to validate the force field for DNA, and I think we have a good idea what works and what doesn't. Again, if you have a specific question, let me know. But my point is that the force field could have an effect. So let me actually elaborate on that a little bit. So how do we know that the force field could have an effect? And so here's an example. We were interested in virus simulations, but from a different angle than Klaus's group. We actually wanted to see what happens to DNA when it's packed in a viral capsid. So here's an image of a virus. You could see that there's barely any room left once you populate it with DNA. So DNA really forms tight contacts. How does it happen? So if you think about virus capsids, although they look rigid, like in this case that looks like a wall, it's actually not. It's permeable to ions and water. So the viral capsid, it confines DNA, but it lets ions and water from the environment to go in and out. So in a way, the ion environment inside DNA can be different than the ion environment outside DNA. So we were curious about what that ion environment is, and also we wanted to see how DNA packs. Like, what is the physics of DNA array? So you can imagine that from a physics point of view, it's kind of interesting to see what happens when you put many charged objects in a confined space and so on. And there have been many interesting papers on the theory side predicting what would happen, whether it has to do with low temperature physics and so on. So lots of theories has gone into explaining the physics of DNA arrays, and we wanted to see if we could simulate it. So what we did was the following. We built a system where we had an array of infinite DNA molecules. So as you can see, I really like those infinite DNA molecules because it simplifies the matter a lot. In this case, we have 64, and they are connected to themselves. All right, how do we model the capsid? We actually don't model the capsid. What we do is we put entire thing into in a confining potential. So what the confining potential does, it does not allow DNA to exit a volume of certain radius, whereas ions and water can freely move in and out. If you ask how do you do it in a molecular dynamics simulation, well, that's actually fairly straightforward. You just indicate which atoms to apply the potential to. But basically, again, so we have this potential that acts on DNA, and because we know how much force we apply to keep the DNA in, by the Newton's third law, that's the force that the DNA applies to the potential. And because we know the radius of the volume, we know the pressure. So basically, this is how we compute the pressure inside of the DNA array. And it will become clear on the next slide why this is important. Again, so the setup is the following. So we have a potential that lets water ions in and out, keeping the DNA a certain distance. And the reason for doing that was that in the 80s, Parthijan and co-workers did a really fantastic job measuring DNA, the distance between DNA and DNA condensate versus pressure. So experimentally, how they do it, they have DNA molecules, a buffer of solution, and PEG molecules. So what PEG molecules do, they induce osmotic pressure. So it's equivalent to the pressures that we apply in our simulations, but basically, at the end of the day, so DNA molecules, they form those kind of bundles. And experimentally, it's known the pressure and the distance between DNA molecules with a high resolution. So for example, those are the data that you would get from the literature, pressure versus distance. And from our simulation system, we can get exactly same quantities, pressure versus distance. The problem was that they were not in agreement by a lot. So when I see disagreement, this is log scale, right? So when the difference is 10 orders of magnitude, I say the force field has a problem. Yeah, if it's like a factor of 2, I would say it's okay. So how do we know it's a force field problem and not our problem the way we did the simulations? So if you look at the way the DNA is organized in those arrays, so each circle is one DNA molecule. So you could see that they form those clusters. And what is shown in the background here is a local ion density. So this kind of light blue color means very few ions, and yellow color means a very high concentration of ions. So basically, you have those local clusters where ions sit, and that's an artifact from a simulation. So if you ever try to simulate a highly concentrated ion solution using standard ion parameters, they will form clusters. They will basically crystallize or semi-crystallize, and it shouldn't happen because from experiments, the solubility limit is 7 molar, and this is like 4. So basically, there was a problem with the force fields that we identified dissolved ions bind to DNA too strongly. So we had to fix it. And let me show you a method to do it. So we fixed it by, again, considering a system that we have good experimental data and we can model one-to-one in our simulations. So here is a system that was proposed by Ru and coworkers to refined force field. So what you have in that system, again, is a semi-permeable wall on both sides of the volume where you have a model compound, in this case like dimethyl phosphate, and our ion of interest. Outside the volume, there's water. So water can pass between the two volumes, no problem, but the ions cannot. So imagine, so you have here ions, and they want to expand, but you confine them. Again, what you measure directly from the simulation is the pressure that the ions apply to the walls to confine them versus ion concentration. So that's what you measure directly from your simulations. And that is also measured directly from osmotic pressure experiments. So there are those old experimental procedures. Probably your chemistry department would have somewhere on the shelf this apparatus that can do it. But basically, you measure osmotic pressure, and then you typically find that your osmotic pressure is wrong. So in this case, what we did, we knew what we wanted to fix. We wanted to fix a particular atom at the phosphate of the DNA. That atom interaction with an ion. So what we did, of course, one way is just to put entire volume of DNA and see what happens, but then it would be very expensive simulations, and we would not observe enough collisions. So what we did, we took a small piece of DNA, actually a DNA mimicking compound called dimethyl phosphate, and we place it here with our ion of choice, for which we have osmotic pressure data. And once we have measured the interactions, then we can correct them by applying a small correction to the van der Waals interaction for that ion pair. And once we do that, we can go back and see if we can reproduce our Parsegens experiment. And indeed, we could. So one more time, how does the force field reparameterization work? You identify a problem. If it's a well-defined problem, like a specific interaction, then you can probably fix it. Then you need to find a system where you have experimental data for that specific interaction, which was osmotic pressure in this case. Once you have that, you go to the force field and you surgically fix it What do I mean by surgically? So in the molecular dynamics force field, there is an option to apply your custom potential to a pair of atoms. So if you do that, you don't break anything else, only that pair. And that's a surgical, because you fix only that pair and nothing else is broken. And this is what we've done here, and it worked very well. So if you are interested in modeling charged systems like DNA or highly charged proteins or lipid bilayers, you probably should use our force field set for ions. It doesn't, it makes it only better. Let's put it this way. Yeah, so I like it. Everything that was there before is there. It's just your ion charge group interaction is correct. All right, I am going to show you last thing, and I will be done here. This is actually the project that the first tutorial is written for. So we were interested in the process of DNA replication and repair. And the DNA divides. And when DNA divides, it has to copy its DNA. And in a cell, the DNA replication is facilitated by, I would call it an orchestra of proteins that work together to to copy DNA. So you start with one DNA and you end up with two. And how it happens, so you have several proteins that coordinate their action, and I'm not going to go into the details. But basically, at some point, you end up with a single-strand DNA that needs to be protected from other proteins that would go and cut it. And that is done using single-strand binding protein, which comes and interacts with DNA. Here it is. So that is a single-strand binding protein, and what is bound to it is a single strand. Yay. All right. So the question was, once a single strand binds to a single-stranded binding protein, how does it come off of it? So imagine if it binds too strongly, then it's kind of game over. Then nothing you can do with it. So it shouldn't bind it too strongly. Is it possible that the DNA diffuse on a single strand? And if it's possible, how does it happen? Right? So those were the questions that we asked. And those were the questions that we also had some answers from our colleagues in the Department of Physics. So Tagjip Ha, he did these measurements with a single molecule fret to investigate different modes of DNA binding. So what do you do with a project like that? I mean, the first thing that you do, of course, you look for the crystal structure, and there was a crystal structure, and that's why we did this project, because if there is no crystal structure, game over. So once you have a crystal structure, you will place it in an electrolyte solution. I think in the tutorial you actually learn how to build this system, right, Chris? Yeah? Yeah? More or less. So, and well, then you have the DNA molecule and the protein, and it tumbles around a lot. I mean, by the way, if you are interested in learning how to make movies and nice-looking images, you can also ask Chris. He's an expert. So this is, I mean, typically if you do it from VMD, it doesn't come out so nice, but you can really push VMD to make fantastic images. All right, so so what did we learn from those simulations? First of all, you'd see that the DNA, it doesn't stay rigid. It kind of diffuses, or I would say binds from the DNA. There's some local dynamics, but okay. So DNA is mobile at the surface of a protein. But the timescale, again, we had the timescale problem because it was just a microsecond, and we couldn't see anything happening in the microsecond, really. So people in our department also used optical traps to pull on the DNA, just like that. So optical traps I already introduced. Remember the pipette and a bead. But in this case, you can be very clever and use the same bead split it into two and use two beads coming from the same bean. This is really fancy single molecule method that basically allows you to minimize the noise. So that's what was done. And in optical trap experiment, you can pull on the ends of the DNA and trying to see the force at which it will come off of the protein, right? And that's what you did. And I think this one is covered in the tutorial. So you basically pull on the ends of the DNA, and you see how the DNA comes off. To make this more difficult, we actually cut off pieces of DNA as it goes, but you don't have to do it. So force versus time looks like that. If you get three graphs like that, I would say there is no correlation. And the reason for that is because the DNA is mobile at the surface of DNA. So basically, it's like trying to enter the river second time at the same spot. So it's always a different river. So once we started the simulations, the DNA moved around, and then once you do force extension curve, you get a different result. So we have to average a lot to make a link with experiment. Or we could do a coarse-grained model, and which is what we did. So we have developed a way to go from all-atom description to a coarse-grained description, which relies on all-atom simulation. And the way we do is, again, is somewhat brute force. We just take a protein and lots of small fragments and see where it wants to bind, like that. So once we know where DNA wants to bind, we can create a local affinity map. So basically, we will represent our protein as a density map, indicating affinity to DNA. Once we do that, then we can use it together with a model of single DNA strands to actually see how it tumbles and runs along. So I think Chris at some point made a tutorial for coarse-grained simulation of those systems. I don't think it's ready for prime time yet. At some point it will be released, and you will find it in our website. So if you're interested in this kind of work, again, please email me. We can make a preliminary version of the tutorial available. But today you'll basically focus on all-atom modeling of those systems. Yeah, here it is. Yeah, so atomistic mechanics of single-stranded DNA binding proteins, that's a tutorial. So you learn how to set up a system like that, and then maybe how to simulate pulling. Are there any questions to this part? If there are no questions, then it's time to get some coffee. All right?